Hi. So this is the problem solving session 9, and today we are going to continue from the topic that we left in the last session. And today we are mainly going to discuss about the nervous system and discussing about the components that make up the nervous system and how does the information is actually relayed. So let's begin. So nervous system, this refers to the characteristic of a living entity wherein it is able to respond to stimuli with the intervention of nervous system. So basically, nervous systems helps a body or an organism to respond to a stimuli. So basically, how you can define nervous system is an organ system that is ascribed to send signals from the spinal cord and brain throughout the body, and as well as receive information back from the body parts. So it's a to and fro motion from the central nervous system, which consists of the spinal cord and the brain, to the body or parts, and then back from body parts to the brain and spinal cord. So, and this information is carried by the neurons. So they act as a mediator, and that's make them the basic unit of signaling in the nervous system. So just to explain how does that relay of information actually takes place. So if you see here, so let's say this eye perceives a stimulus. Then there are sensory neurons which pick up this cue and gives information to the brain. The brain then sends a message to the motor neuron, which goes to the hands, for example, and that says it to pick up the glass. So that's the kind of information that is to the brain and from the brain. Another kind of information, for example, here you can see that this person is touching the source of a heat, so they're touching the candle. So what your reaction should be is to remove your hand. So when you touch the flame, the receiver, so that's carrying this response, it takes that to the spinal cord, and the spinal cord then responds and sends a message to the arms saying that remove that hand. So that's the kind of information that is being relayed. So moving forward, so nervous system, they consist of this complex network of neurons. Neurons are the basic signaling unit, and there are multiple of these neurons in the network that carry the messages. And these nervous tissue, they originate from the ectoderm, and they specialize for the this purpose of receiving stimuli and then transmitting the message. But the kind of nervous system that are present in different organisms are different on the level of complexity. For example, here you can see in hydra, it is just a very simple nerve net. So all these, there will be a basic signaling center, and then there will be these nerve net that spread throughout the body. But then when you go to crab, you see that there is more complexity with it having two ganglions. So ganglions are nerve centers. So one will be present on the dorsal side and one will be present on the ventral side. And those are the relaying centers. And the most complicated you can see in the vertebrates, where you have billions of neurons connected to the ganglions and to the central nervous system. Other than that, even in sea stars or echinodermata, you can see that they will have these radial nerves originating from a single nerve center. In flatworms, you have nerve cords. In bees, you have the ganglia, which is in the brain, which is relaying the information all throughout the body. And in the octopus, you can see that there are several ganglia at each side. So you have brain, then you have the nerve spreading to eye and then to the each tentacles. So coming to human nervous system, as you can imagine, there are multiple networks of these neurons, and these neurons are billions in numbers. So these are all interconnected, and the main relaying centers are the brain and the spinal cord. So these neural system here integrates the activities of the organs. So all the organs and all the processes means the fingers, the toes, and every part of your body is connected by nerve. And all those nerves are interconnected, and they are also connected to the central nervous system. They're all there to receive a stimuli and then transmit the information and then get the information back so that they can appropriate response can be given. So, and these messages that are transmitted, they are in the form of electrical impulses, and these are, again, they are carrying the messages from sense organs in to and fro motion. So if you have to see the participating organs in nervous system, they are the sense organs, nerves, spinal cord, and the brain. So as you can see in this diagram here, so you have the brain, you have spinal cord, and then you have so many nerves at different points of the body. So for example, there in the lumbar part, you have the lumbar plexus. Then you in the legs, in the tibia, you have tibial nerve, you have the peroneal nerve, then you have the iliohypogastric nerve. So every place has a dedicated nerve to receive signals. Now, to define it more, the nervous system can be divided into several layers. First is into the central and the peripheral nervous system. So, the central nervous system consists of the main relaying center, which are the brain and spinal cord. Peripheral nervous systems are the nerves, which carry the information to the different parts of the body. They are further subdivided into autonomic and somatic. So, autonomic are those nerves that can control the organs and glands. So, these are involuntary activities. But the somatic are the ones which are for the control of skin and muscles. So these are the voluntary actions that one can control. So the nerves that control these kind of motions are the somatic nerves. And for the autonomic, which are for the organs and for the involuntary activities, they are further divided into sympathetic and parasympathetic. Parasympathetic, basically, again, these are also working on the internal organs and smooth muscles and glands, but these have opposing effects. One of them will be for more arousing, so basically they will be initiating or enhancing the certain activity, while the parasympathetic will be calming those activities down. And we'll see when we come to the later slide what exactly do these nerves carry out. So let's start with central nervous system first. So central nervous system, as I have mentioned, they consist of brain and the spinal cord. So you can see here that they are connected by in the long running process. For example, here you can see this is brain, and then in the continuation you have the spinal cord. And we'll go into detail of both of these structure in the coming slides. So we'll start with brain first. So as you know, as you are told multiple times, that brain is one of the most important information relaying center in your body. So, and it is also a largest organ in the human nervous system. So it is a control unit which helps us. As you know, brain can discover new things, remember things, understand and make decisions. And all these happens via the use of the structural components of brains, as we will see in the slides. So the first thing is that you have a skull, and the brain is enclosed in the skulls for protection, and that is protected from the frontal side. So frontal being the and like this part, lateral being on the sides, and dorsal being on the upper side. So for frontal, lateral and dorsal protection, the brain is enclosed in the skull. And if you see here, so these are different axes of the brain sections. So this is a lateral view. So this is from the side. Superior view is from the top. If you see the brain from the top, how does it look? Then sagittal is at the lateral view. If you have a plane of dissection, if you divide it into two halves, how will that interior part look? And inferior being the lower part. So you can see how complex the structure of brain is, and you can see how different components come out. If you see, so there are many components which are hidden, so it can only be seen if you see from different views. So the brain in its covering has three layers, and which are called the meninges. They are durameter, arachnoid and pyometer. So durameter being the outermost layer, it is strong and it is non-elastic in nature, and it is made up of collagen. Arachnoid is the middle layer. They are thin, and you will see them having highly folded structure. And the last and the innermost layer is the pyometer, which is a thin, delicate and highly vascular nature, and it is what is stuck to the brain. It remains firmly adhering to the brain. So here you can see the layers here. So this one is the bone. So this is the skull bones that we are talking about. Then you have the outermost layer, which is the durameter. Then you have the arachnoid matter here, this layer. And then very internal to that, you have the pyometer. And among the spaces which is between the two layers, you have these kind of spaces which are filled with fluids. So that's what we call. So if it is between durameter and the arachnoid, this is called the subdural space, which is filled with serous fluid. And the space between the inner layers, which is the arachnoid and pyometer, it is called subarachnoid space. For example, here you can see this. And this is filled by the cerebrospinal fluid. So in short, it is called generally CSF. So this, what does CSF do? We have read about lymph. So they're similar to lymph. This is also clear and alkaline fluid. And it gives you the volume. So it gives you protection to the brain. So that's the main function of these fluid. They give support to the brain so that it can protect itself from any damages. And along with this, they also help in exchange of metabolic substances between the brain and the blood capillaries. So this is where the blood capillaries will exchange its nutrients with brain. So CSF is what receives the nutrients and gives out the nutrients to the gives out the materials to into the blood. And CSF is present in the ventricles, subarachnoid, and spinal cord. So it is, so you can, if you imagine, it is present in the interior parts of the brain. The brain is then further, you can divide it into three parts: the forebrain, midbrain, and hindbrain. So basically, in the order of how they are arranged, you can arrange them as forebrain, then the middle portion being midbrain, and then the later it is the hindbrain. So if you see here in this diagram, this part is for forebrain. It consists of cerebrum, thalamus, and hypothalamus. And then you have the midbrain, which is here. So this connects the brainstem to this like this brainstem is connected to the forebrain by the midbrain, and it consists of tectum and tegmentum. And at the end, you have the hindbrain, which consists of cerebellum, medulla, and pons. And this further continues into the spinal cord. So we will begin with forebrain. So forebrain consisted of, as we had seen last time, that it consists of cerebrum, thalamus, and hypothalamus. So first is cerebrum. So cerebrum is the most advanced part of the brain. And why do we see advanced part? Because it consists of multiple lobes which has different functionality. We'll see in a while. And it is also the largest part of the brain. And this consists of two hemispheres. So these are called cerebral hemispheres, and it is on the dorsal surface. So in the upper side of the surface of forebrain, you have two hemispheres which are called cerebral hemispheres. So this as you can see here, that this is the cerebral hemisphere. So this is a sagittal view. So if you see from the upper portion, it is divided into two parts, and these parts are connected by corpus callosum, and in between of these hemispheres there is something called corpus callosum. This corpus callosum is basically, they are nerve fibers which are interconnected between the two hemispheres. So to understand that better, if you see here, so what you are seeing here is one part of the hemisphere, the other part of the hemisphere is towards you. So both of these are connected at this side, which is called the corpus callosum. And fissure is this part, just in between of these two hemispheres. So the outer portion of cerebrum is called the cerebral cortex here. And cerebral cortex makes up of multiple cell bodies, and they have few myelinated axons. Basically, they have most of cell bodies which carry out the information. And that is why they have this gray material, because myelins are sheets which are white in color. So that's why, because they have less of myelinated axons as we know, they relay the informations. So they have no myelin sheath on them. So these are cell bodies which will look gray if you look at them when you're looking anatomically. So if you see here in the picture, all these dark colored sections are the axons, and these are called gray matter. These are outside. And then in the inside, you have these material which are called the white matter, which will have myelinated axons. And so this makes up the cortex, and as you can see, they are highly folded. So here, if you look at the upper side, so in these folds, if they are coming towards the upper side, they are called gyri, and if they are downward grooves, they are called the sulci. So like I mentioned, beneath the gray matter, you will have the myelinated axons. Myelinated axons will have, there will be less cell bodies and more of these myelinated sheath axons, which will give them a white appearance. So that's why they are called white matter. So there is gray matter and white matter. Gray matter on the outside and white matter on the inside for the brain. Further, as I had mentioned, that this is a very advanced part of the brain. So you can see that each cerebral hemisphere, so each hemisphere, okay, so whatever you are reading here, it's in double. So whatever we see here is present on the other half as well. So the hemisphere can be divided into 4 lobes. So you have the frontal lobe, then you have the temporal lobe and parietal lobe, and then at the end you have the occipital lobe. These are also called frontal or anterior lobe because they are present in the anterior side of the brain. Then you have the parietal, which is in the middle layer, so it's called the middle lobe. Then you have the temporal or lateral because it is on the side. So it is here. So it is on the side of the brain. And at the end, you have the occipital or the posterior. So because as you can see, it's at the end towards the rear side of the brain. Now, so these lobes are separated from each other by different these layers. So different networks of nerves and different parts, structures. So for example, the frontal lobe here and the parietal lobe here, they are separated from each other by central sulcus. Then you have the frontal lobe again, but they are separated from the parietal front. So the frontal lobe and parietal lobe are different. So they separate from the temporal lobe based on the sylvian sulcus. So there are different parts of these nerves which are responsible for differentiating these separate lobes. And then you have the occipital lobe and the parietal lobe being separated by these parieto-occipital sulcus. Now, moving on, these different lobes have different functions. So, if you just try to understand, all these lobes will have different nerve centers, and they will respond to different cues. So, if you see something for memory, one of the lobe which is responsible for it will get activated. Similarly, if you see some auditory cues, which is for ears, then you have several lobes which get activated in the brain. And that's how you divide the cerebrum. So you have some region which is for sensory area, some for association, which is for mainly for memories, and then you have one region which is for motor area. So that's what it receives, all kind of information. So ranging from auditory, visual, somatosensory, which means that any touch or any sensation via your processes, and connect them with the cortices. So here you have olfactory bulb, and that process the olfactory information, and then so all this information have to be relayed to the center. The center is generally thalamus. Is only the olfactory cues are sent to the olfactory cortex instead of thalamus. Rest every such information in terms of cues are sent to thalamus for appropriate response. And cerebral cortex are also responsible for speech, language traits. And then you have the hippocampus, which is responsible for memory. So just to give you a overview of how does this function, so you can see that this part, so there are several parts of these hemispheres, and you can see how they are responsible for different activities. For example, here you have the primary motor cortex. They are responsible for the voluntary movements. These premotor cortex, which are just anterior to it, they coordinate with this cortex so that they can they can together control the voluntary movements. Yeah. Then you have so these are the prefrontal association areas. So as I have mentioned, they also have association areas. So this is the prefrontal areas, and these are responsible for the ideas and plan, which is for voluntary movement. So what does it mean? So if you have something to do, for example, lift something or touch something, you first make a plan for it. You first design a thought to it. So all these are present in this, or all these are carried. The information is relayed to this area. Then you have this little Broca's areas. This is responsible for speech. This is the olfactory cortex. So this is where the olfactory cues are received, and the response are also mediated by this the region. Then you have the limbic association concepts. Limbic means emotions, learning, memory. All these are taken in this area. Then you have the primary auditory cortex, so which is what receives the auditory cues, and all these are associated areas of the auditory cortex. Then you have something called Wernicke's area, which is for language comprehension. So you can see what you are hearing and how you are comprehending is also being regulated by brain, also being affected by brain. Then you have visual cortex, which is for vision, and then you have the associated areas for the visual processing. And then lastly, you also have for the sensory area. So these are the sensory areas where you have the somatostatic information being relayed to. So that's why I just mentioned that how cerebrum is the most advanced part of the brain. So moving on from cerebrum, the next part is diencephalon. So diencephalon is somewhere here. So if this is cerebrum, this part is the diencephalon, and this connects the brainstem and the cerebrum. And again, it also acts as a relay and processing center for the sensory and autonomic information. Next, you have the thalamus, the main relay center where all the informations are taken to. So these are here, present in the anterior part here, and they accept all the sensory impulses of the body except for the olfaction, because olfaction goes to the olfactory cortex. And in some organisms, especially lower organisms, this is where the sensory center mainly lies. So not the cerebral cortex, which has all these different areas, but it is the thalamus that receives all the information, because that's how that is. Like, the cerebral cortex is not that well developed in those organisms as compared to the higher organism. For example, us, we have one of the most developed cerebral cortexes. Okay, then moving on, the next part is hypothalamus. So hypothalamus is this side. So if you see here once again, so this is the cerebrum. Here you have the pituitary gland, and if you take the pituitary gland, zoom into it, you will see that they have anterior pituitary and posterior pituitary, and this is the hypothalamus right above it. And this is important because you will read about many hormones being secreted from the pituitary gland. The hypothalamus receiving the cue to actually help in the secretion of these hormones. So that's why it is so important a structure in the brain. So, and that's why it's also called the master gland. And because of this regulation of secretion of hormones, it is responsible for maintaining the body's internal balance, homeostasis basically. And it also, that's why it connects the endocrine. We have read about the endocrine, how endocrine is responsible for the production of hormones, and it connects nervous system and endocrine. So they receive cue from the endocrine saying that, okay, so let's say the sugar has so you need glucagon to synthesize your sugar. So glucagon information is relayed by the nervous system from the endocrine, and then hypothalamus as a response synthesizes glucagon. It initiates a signal to synthesize glucagon from the respective gland, and then the action is further carried out. Beyond hypothalamus, you also have two other parts, which is called the epithalamus and the metathalamus. These also have some very specific role. For example, here you can see that the epithalamus has something called pineal gland, and pineal gland is responsible again for important characteristic, like for example, just a second. Yeah. Yeah, so pineal gland, as I was mentioning, again secretes hormones and maintains some important functions of your body. And then again, metathalamus, they are also they consist of this something called medial geniculate body. They are responsible for the conduction, or they're related to vision and hearing. So they are mainly controlling the vision and the hearing aspects. Now, moving from forebrain, we move to the middle portion, which has the midbrain. So it is in, if you have to just simply say, they connect the hindbrain to the forebrain. Here they consist of the brainstem. So they are part of the brainstem. Brainstem being the lower end of the brain. So brainstem, and they develop from mesencephalon. So it contains relay nuclei being a center of two cranial nerves. And these nerves are oculomotor and trochlear nerves. And as you can, just by name itself, you can see that trochlea being the part of your auditory canal, and oculomotor part of your visual information. So they are mainly responsible for the processing of auditory and visual information. And they also regulate the eye and eyelid movement. So your any movement, any involuntary or voluntary movement is regulated by the parts of brain and spinal cord. So you can see how everything is well connected in our body. So the main part of the midbrains are the tectum. So these parts, these regions here. Then you have the cerebral aqueduct. Then you have the tegmentum, and then finally you have the cerebral peduncles. And these also, of course, will act as centers for visual and auditory informations, and together with the brainstem, they will control the autonomic function. So they are responsible for mainly controlling the autonomic function, which are the involuntary activities of the organs and the glands. Next, we move on to hindbrain. So hindbrain is the lowest part of the brain, and at the end, so towards the rear side. And these are called rhomboencephalon. They are further divided into two types. This is called metencephalon and myelencephalon. Metencephalon will give rise to cerebellum and pons, and the myelencephalon will give rise to medulla. So if you see here, These are the two encephalons. Encephalons is a region in the brain. And to divide them further, you have the pons on this side, cerebellum here, and medulla at the end, just above the spinal cord here. So medulla is also called the medulla oblongata. Now in pons, this is mainly responsible for the sleep-based cycle. So your circadian rhythm is mainly controlled by the pons, and also the dream center. So if you see any dream, this is because of pons getting activated. So here you also have the pneumotaxic center, which regulates the inhalation and exhalation. So you can see now that even a simple motion like breathing is regulated by a part in the brain. Then you have cranial nerves. Cranial nerves means nerves which are of the brain. So cranial nerves, there are four of these present in this region, and they are responsible for several involuntary actions like bladder control, for urination, for hearing, what kind of information you can hear, for your postures, for chewing, swallowing, taste. So all these are motions which are associated with the mouth. Then you have for even for taste, then facial expressions and sensation. These are all part of pons. Moving on to cerebellum. So here you have cerebellum. So cerebellum is responsible for coordination and fine tuning of all the motor activities. So any movement with your muscle is responsible by the cerebellum. So cerebellum regulates these kind of motions. And again, so they will also be regulating the equilibrium of the muscles, postures, and the muscle tone. And how is this important? Because all these muscle activities which you do, for example, in the sports like swimming, cycling, all these involve muscle activities, and these are controlled by the cerebellum. And lastly, you have medulla. And medulla controls mainly the autonomic functions, for example, respiration. And then you have the cardiovascular reflexes. This we have read in the last session as well. And for example, others like swallowing, vomiting, and coughing. So all these involuntary active actions, which are the autonomic functions, they are controlled by the medulla. Now moving on to spinal cord. So in continuation from the end portion of the brain, you have spinal cord, which is in running throughout your body. So this is so for example, you can see here that this is long pipeline structure which arises from the medulla oblongata, and then it runs across the vertebral column of the backbone. And then it is further segmented into several regions based on which area of the body it is part of. So for example, the anterior portions are called cervical, then you have the thoracic, then at the end you have lumbar, and at the very end you have the sacral or coccygeal. And talking about nerve fibers, they are segmented with a pair of roots, which is the dorsal and the ventral. So dorsal means the upper, ventral being the lower part, the belly side. So belly side is called the ventral, and dorsal is the upper part, or the back side, if you have to tell. So they have roots at both these sides, and these join to form the spinal nerve. So in the brain, the nerves are called cranial nerves. In spinal cord, they are called the spinal nerves. And so when it is continuing from the medulla oblongata, it comes out as into the foramen of magnum. So this foramen magnum is where it comes continues into the spinal cord. And this too is supported by the dura mater, arachnoid, and pyometer, just like how it was for the brain. So if you have to see it structurally, you can see then it has three layers, which are the dura mater, arachnoid, and pyometer. Now something different here. In brain, you have gray matter outside and white matter inside. Again, just to reiterate, gray matter being the non-myelinated axons, white matter have myelinated axons. Myelinated means having myelin sheath around them. So in brain, you had these gray matter outside and white matter inside. In spinal cord, you have the opposite. You have the gray matter inside and white matter outside. And in the root of these dorsal horns, in the dorsal side, you will have the dorsal root ganglion, as we have just mentioned. And here you have sensory neurons whose axon extend and then get embedded into the gray matter. So from the ganglion are basically collection of nerve fibers. From here, the nerve fibers differentiate into different parts of the brain, different parts of the system. So sensory neurons are part of the dorsal root ganglia. So all the sensory motion that is received as a cue for the spinal cord are received in the dorsal ganglion. While for the motor neurons, the ones which are actually carrying out the activities, those are motor neurons, and those are found in the ventral root. Cyton means the body, cell body. So whose cell body is found in the ventral horn, and the dendrons get embedded. So you see the structure is this. You have a ganglion here, you have a ganglion here. Both of these affect out the dendrons, and they get connected over here in this structure. So so the roots, they give out these nerves, which are the dendrites, and these come and can get connected into the main part. So both these sensory and motor nerve fiber combine, and then they are, so when they come out as spinal nerve, they are both, they consist of both these dorsal root ganglion nerves and the ventral root ganglion nerves. Uh, and they act as a bridge between the brain. So brain gives an information that comes to the spine, and then spine gives it out to the body organs. So as a result, they regulate and conduct the reflex actions and the several impulses which come from the brain. So this is about central nervous system. Now we have read about how information is relayed and how they are, the brain and the spinal cord perceives the information and then regulates and then gives out the information that is required to, or this is appropriate for the response. Now, but who actually carries the information? It is the peripheral nervous system. So these are all the nerves across the body which carries the information. So that's why they are called peripheral. Peripheral means being on the edges. So peripheral nervous system. So if you see here, peripheral nervous system, they develop from central nervous system, but they go over to get connected with different parts of the body. And because they are responsible for carrying out, because they are receiving information from all the parts of the brain and the spinal cord, they control both the voluntary and the involuntary actions. But they do so on the basis of the different nerve system. That's how they get divided into autonomous and somatic. We'll come to that. So when you have, so that the nerve fibers again here can be divided into two fibers. What is nerve fibers? So basically how, so the direction of the relay of information. Based on that, nerve fibers can also be divided as two types. So they can be called afferent nerve fibers or efferent nerve fibers. So afferent means, so they transmit the information from the tissue to the CNS. So afferent means on the flow. So they receive information, take it to the CNS. Efferent is effective, so they bring out the effect. So that's why they carry the information from CNS to the corresponding peripheral organs. So information to the CNS is carried by afferent nerve fibers. Information from the CNS is carried by efferent nerve fibers. And as you can see, so as I mentioned that different parts have different nerve fibers. So you can see here in the brain you have the cranial nerve, at the end you have the vagus nerve, and then you have different parts of your body. For example, radius, ulna, media will all have their own nerves. Then in the legs, you have something called sciatic nerve, which is on your thighs. Tibia has tibial nerve. Then you have all these nerves which connect. So in one region, you have different nerves which are connected to different regions of your muscles. So you can have common peroneal nerve, then you have deep peroneal nerve, and then you have superficial peroneal nerve. So you can see how at a simple region also you have so many types of nerves. And then for example here for spinal cords, you have, this was the initial part was called cervical. So cervical nerves, which are 8 in number, they all get connected to different parts of the region. So you can see the first one gets to head and neck, second one goes to diaphragm, then deltoids being in the shoulders and the bridge biceps. Then you have wrist extenders. The one of the nerves specifically goes to your wrist so that you can extend them. Seventh is goes to triceps, and the last one goes to your hands, basically your palms. Then you have the thoracic nerves, which are 12 in number, and they are mainly, so thoracic is your middle portion, the chest and abdomen. So these spread all across your chest and abdomen. Lumbar, it goes to your legs. And lastly, you have the sacral nerve, which goes to the bowel and bladder, and also for the sexual function. So like I was saying, so you can divide your peripheral nervous system into two types, which is the autonomic and the somatic nervous system. So somatic neural system, as I had mentioned in the beginning, this is for the voluntary action. So all the skeletal muscle information which involves you to control your hand. So like you can move your hand, you can move your leg, you can move your body. So all these skeletal muscle activities are controlled by the somatic neural system. And that's why they have the somatic nerves. But the ones which are for the glands, for the organs, for the smooth muscles, which are involuntary action, they are for the autonomous neural system. And these are self-regulating system, and these can be then further divided into sympathetic and parasympathetic nervous system. So what does it mean to have, like, what does parasympathetic and sympathetic nervous system do? Together, they control the involuntary actions of the body, for example, heartbeat, sweating, peristalsis, and they consist of motor neurons to carry out the information. Sensory receives the information. And so, broadly, if you want to talk about these two nervous system, sympathetic relates to intuitive reaction, which increases the protection of the body, while the parasympathetic is where you conserve the energy, so you cool down the body. So here, if you see the functionality, in sympathetic you will have dilation of pupil. When do you dilute pupil? When you, let's say, you get stressed, or you see something exciting, or you sense, or you get scared, so you react and your that pupils are dilated. So that's the kind of responding to your stimuli. So when you're responding, sympathetic nervous system will come into play. So your pupils get in dilated, you have saliva which gets inhibited, your bronchia gets dilated, your heart rate increases, and then at the same time you have the inhibition of digestive organs and pancreas and the gallbladder. Moving down, you have the sympathetic system stimulating the adrenal medulla to release adrenaline and non-adrenaline, and these are the fight or flight response hormones. Then you have relaxing the unibilary bladders, and then you have these also work on the genitals. But then you have, in contrast, the parasympathetic, which will work to reduce these effects. So in contrast to dilating pupil, it will work to constrict the pupils. It will increase the saliva production. It will reduce the heart rate, bring it back to normalcy, constrict the bronchia, and then all those activities in the digestive organ which was being inhibited will be stimulated by the parasympathetic nervous system. So you see, they work in consort but in the opposite direction to maintain homeostasis. Now, all these was about a nervous system anatomy. Now we will study about how nerve conduction. So we'll study nerve conduction, and we'll see how studies of the nerve conduction helps in identifying several diseases. So you would, if you would have undergone any nerve conduction study, you would have this kind of setup. So what does this is a medical setup. So what happens here is that they use it to estimate the capability of the electrical conduction and of the motor nerves and sensory nerves of the human body. So how well your motor nerves, your sensory nerves, or your nervous system basically is functioning. To understand that, this kind of study is carried out, which is called the nerve conduction study. Now, how does this work, Sean? This measures the electrical impulses and how fast they move. So there is a certain expectation that within certain limit, your nerve impulses should reach the distant location. And this is what you try to see in your nervous system, that based on the study, does your body is functioning normally. And the output is generally electromyography. So electro means electrical activity, myography means related to muscles. So you are looking at the electrical activity of your muscles as well, along with the nerve conduction study, because nerve conduction is affected by muscle movement. So you have a good readout to do. So you can check the muscle movement and you can try to interpret the nerve conductions. So both these together help you to identify the presence, location, and the extent of diseases if there is any damage to nerve and muscles. So how does this study actually happen? So you have this kind of setup here. And so what happens is you stimulate a nerve. And it is done with you. So you have these electrode patches, which is connected, right? So when you stimulate a nerve, one of the electrodes gets stimulated with a very mild electric impulse. And as and when that electrode is stimulated, the other electrode records it. And then the resulting electrical activity is, because it is recording it, that is recorded by another electrode. And this is continued. And at the end of it, what you try to see is how fast this information was relayed. So and how do you calculate that? Because you know a certain distance is there between your nerves, and you have a certain velocity which is being carried, like at a certain time it has happened. So you find the velocity of the conduction by calculating the distance it traveled given the certain time that it took to cover that much of distance. Simple, like you calculate the velocity by distance by time. So that's what we try to do here as well. The time taken for the impulse to actually conduct or relay is called the latency period or latency. So, based on what kind of nerves you study and what kind of information, which part of the nerve you're actually carrying information from, it is again called motor nerve conduction study or the sensory nerve conduction study. Again, motor being something which actually carries out information, sensory is what receives the information. So, if you have a motor nerve conduction study, it is performed by the stimulation of a peripheral nerve and but recording from a muscle to which these supply. So these are motor nerves, right? So they will supply the information to one of the muscle. So you take the information from a muscle, and here the latency is again defined as a time taken for the electrical impulse to travel the distance from the stimulation to that muscle, and the time being in milliseconds. But if you have a sensory nerve conduction study, this again is done by the electrical stimulation of a peripheral nerve, but in contrast to a muscle where it is supplying, for example, in motor nerves, here in sensory, the recording is done from actually portion which is sensory of the part of the nerve. Basically, the one which is sensing, which is responsible for actually sensing the cues. So that part you are they're checking the information. So you record from that region, and then you carry the same formula to calculate the velocity. So this was based on where you put your recording electrode at, but the type of study and what kind of study, like what location you are looking at, what region you are trying to understand, based on that you have these different kind of study. One is called F wave study and one is called H reflex study. So F wave study means where you are trying to look at the velocity between the limb here and the spine. So in this, you have a strong electrical stimulus which is applied at the distal portion of the nerve, and the response or the resulting impulse, it moves in two directions. One towards the muscle fiber, basically the limb, and the other towards the spinal cord. So you have the spinal cord and the limb system both being part of this study. But the other study, which is again H reflex study, which again carries quite between the limb and the spinal cord. Here, the impulses are going towards the spinal cord and they are sensory nerves, while the impulse is coming from the spinal nerve. So the nerves which are involved towards the spinal cord are different. In the F study, you are mainly category or choosing to look at the motor nerves, but in the H reflex, you are looking at the sensory nerves and the motor nerves both. So that was about nerve conduction study. Now, other than that, you also can look at the activity, electrical activity of the brain. How do you do that? So these are two graphs that I am sure, if not the left one, you would have at least know about the right one. You would have seen this right one in some of the medical forms, or sometimes your test would have been done. So you can guess what could this be. One of them is called EEG and one of them is called ECG. The ECG is for the cardiograph, so something related to heart, and the EEG is related to brain. So on your slide right now, you have two activities graphs. One is for the brain and one is for the heart. Can you take a guess on which one could be heart and which one could be muscle based on what you have seen for now? I'll give the cue in another 5 seconds. Yeah, so on the left of your screen, it is the brain activity. So this is called the electro, because we are looking at the electrical activity, encephalogram. So this is from the brain. While on the right, you have the ECG, which is the electrocardiogram. So this is heart activity and this is brain's activity. And you can see how similar they are. They are both wave functions of the electrical conduction. And we read them, or we look at their activity and pattern to understand how well they are functioning. So moving on to EEG or electroencephalogram. So this technique was discovered in somewhere around 1930s by Hans Berg, and this is a technique to record and interpret the activity of the brain, and you look at the brain waves. So what do you do in general? So you can see this is a setup for a EEG where you have all these different nerve electrodes connected to different nerves in the brain. So the nerve cells here generate the electric waves, and these waves are then recorded on that screen here, and then these give out the graph. and then you interpret the graph of how are they happening. And this generally takes place if you have brain tumor or any head injury, or even if you have sleeping disorder, for if you example apnea, sleep apnea you have, or any infections or anything related to the cerebrum or any part of the brain, or epilepsy, or even if you have brain death, that can also be looked by the activity of the brain. So what happens here is, again, it is based on the principle of volume conduction. So you have these electric potentials. We'll see about this in a while. So when your nerves get stimulated or your brains get stimulated, they generate electrical potential or action potential. And so because your brain consists of multiple neurons here, all these neurons, they maintain a certain resting potential. And when they need, they generate an action potential. And this action potential, as we will see, this is due to the difference in the concentration of ions. Now, when you are stimulating your brain during this study, a large number of ions will get expelled from the neurons, and they will generate this action potential, which will lead to the release of these waves, which will be carried forward in the brain. And because you have electrodes connected to different parts of the brain, as and when the wave reaches the electrode, the pulse the electrons, they will push the electrons, and because the electrons will be pushed, this will be recorded as a voltage difference, and the difference in the voltage is what you see in the graph, and that is directed by a voltmeter, and that will give out the output as how was the drop and how was the rise in the voltage. And that's how you see it here. So you see the amplitude. So based on what kind of information, or like what information you receive from the voltmeter, you can generate the amplitude. How amplitude being the length of the wave and the distance of the wave. So that can be looked into by the information that is received. So there are different kind of waves that are generated by the brains at different time of your cycle or the day. So we'll see. So for example, one of the waves is delta wave. So they range from 1 to 4, less than 4 hertz, and they are, so if you want to detect some subcortical lesions or some midline, deep midline being the fissure, so something related to cerebral part, you basically look for these waves because these kind of impulses are mostly propagating there. And if you see here, these are basically the delta waves, and as you can see, they have highest amplitude, and because they are highest amplitude and they have this broad length, they are the slowest wave that will be propagated. The next in line is theta wave. They have a range of 4 to 7 hertz, and this is mainly in the young children. So when some young children are being studied for EEG, the main wave that is looked into is the theta wave. And there they were used for the focal subcortical lesion detections, or again the midline disorder. So all these delta waves, which were the activity that was being looked by delta waves in the adults, are looked by for theta wave in the young. Then you have the alpha waves. These are abundant in the posterior region of the brain. So anything which has got to look into the posterior region of the brain, alpha wave will give the information of how well it is. So it is associated with the inhibition of elicited response. So now that's what I was mentioning. So if you are awake, but you are a resting person, basically if you are just in the trying go to sleep, you generate alpha wave. So if you are trying to sleep but you're not asleep, but you are resting, then your brain has all the alpha waves which are in motion, and that's what happens in coma patients, because they're not awake but they're just resting, so they have most of the alpha waves in their posterior brain, and mainly they are propagating in the posterior region. Next is beta wave. So you have beta wave which are in the frontal region. So the posterior you had the alpha wave, in the frontal region you have the beta waves, and you can see here they have symmetrical distribution and low amplitudes, and these are associated with high thinking and anxious behavior. So if you are alert, if you see something and you get alerted, these waves propagate faster, or these waves are generated at a greater frequency, and because they are responsible for relaying the information for the alertness. Next is gamma wave here. So this is responsible for decline in cognitive ability. So if you can't recognize something, or you have some problem with your cognitive ability, it is because gamma waves are in action. And the last one is something called mu wave. These are mainly for the sensory motor. So sensory motor means involving the senses and the motor neurons, and they are responsible for the firing of motor neurons in rest state. So all these are different waves, which when you look into the final output from the study of EEG, you can see if one of them are not behaving as per expectations, you know that that part of the brain is somehow not correct, or it is being that is the part that is affected in that particular patient. So now all these waves we have studied and all these anatomical structures we have studied, so we'll combine them together and we will try to see how does the nervous tissue actually function. So this is how nervous tissue looks under the microscope. And so these long parts that you see here, they are the dendrites. We had seen part of dendrites in the previous session, but we will see how dendrites actually function and look in coming slides. You have the main structure here, which is the cell body with the nucleus, and then the axons, the main length which is carried through the cell body. And then you have the neuroglia, so the glial cells, which are associated with the neural cells. And these together make up the nervous tissue. So their main function is, of course, to monitor and regulate the function of the body. They have the nerve cells and the glial cells. Nerve cells are called neurons, and you have brain and spinal cord all composed of nervous tissue. Nervous tissue means all these neural cells packed together. And just to say again, they are the stiff tissue component of the CNS and the PNS, central nervous system and peripheral nervous system. And they are specialized cells that react to stimuli because of the presence of these dendrites here. So these dendrites here pick up the information, relay it through the axons, and then take it to the destined locations. So these are neurons, the basic unit. So they have this cyton, the main cell body, and then the cell process. Cell process is the extension of the cell body. So the cyton here, the first cell body, you can see here that they have this broader part which consists this uninucleated cytoplasm. So there is one nucleus in the cytoplasm, and everything that you find in a normal cell, all the organelles, mitochondria, Golgi bodies, they're all present here except for the centriole. Now, what was centriole actually doing? What is the actual role of centriole? Could you remember? Try to remember. So centriole was part of cell division. When we were reading cell division, we had come across this term centriole. So now you can see that centriole is responsible for cell division or helps in cell division. And because there is no centriole in the cyton or the neurons, you can make a guess here that these cells do not divide. So these cells basically are the non-dividing cells of the body. Okay. Now, in the cell organelles here, you have neurofibrils and you have something called Nissl granule. And these are mainly responsible for the impulse transfer. And Nissl granules are basically the endoplasmic reticulum, which are highly coiled, which are present in these cell body. So just to go over the cell body once again, you have the Nissl substance here, then you have the mitochondria, and then you have the neurofibrils. So fibrils basically are this long tract where the information can be carried forward. And then at the end, you have the axon coming out, and they mainly come out from this region, which is called the axon hillock. And then you have this long process, which ultimately ends into dendrites. These could be covered with the myelin sheath or not. If they are covered with myelin sheath, this layer is called myelin sheath, and the interjunction, the junction between two myelin sheath is called the nodes of Ranvier. And this myelin sheath is covered by a squamous shell. So that's what we are mentioning here as well. So the cell process here, they consist of the main region which is called the axon. These axons can have myelin sheath or not. If they have myelin sheath, they are mainly constituted by these squamous cells, one squamous cells, and the interjunction is called the rods of Ranvier. And the dendrites, which is at the end, the highly branched section here. So these are dendrites. So these are the sensory cues. So they receive the information from other neurons and synapses. And the end, you have the axon terminal, and we had seen the axon terminal relaying information to the next neurons. So you will have dendrites here of other neurons, and there's information are carried to those neurons afterwards. So yeah, so what is myelin sheath? What do we mean by myelin sheath? So you have these myelin sheath made up of sphingomyelin, which is a phospholipid, and they act as insulator. Basically, they prevent leakage of ions. So what happens is, in some parts of the brain, you need them to be protected, the information to be carried. So there, to avoid the loss of information, these myelin sheaths are present on the axon so that the ions are not lost. We will see why losing iron is important, or why the ions are so important for the electrical impulse. But just to not let go of the information or not to lose the impulse, there is myelin sheath which acts as an insulator. And most importantly, information in neurons are carried in one direction only. So it passes from the neuron, from the dendrites through here. So it's always the dendrites that will receive the information, and then the information is carried via this long branch. Okay. And also, the sconce cell, I forgot to mention that sconce cells are also called the neurolemma. So the layer which is surrounding the myelin sheath is called the neurolemma. Okay. So based on the type of dendrites, where the dendrites are present and where your axons are present, there are different kind of neurons that are present in different kind of cells. So unipolar neuron, as you can see, that has one pole, means one end. So you have only one process, which is one axon from the cyton or the main cell body. And you find this in nervous system of embryo especially. Sometimes the neurons, see the axons will be again divided. So you have this process which divided into two parts. So this process here, which was actually arising here, it got divided into two parts. And then here, one of them will act as axon and the other will act as dendrite. So what happens here is that your cyton is here. So originally, if you see, this is the cyton, this is the axon, and this would be your N terminals. But here you have the cyton here, and then you have two parts coming out from it. One is acting as an axon, and one is acting as a dendrite. These are then called the pseudo unipolar neurons, and these you will find in the dorsal root ganglia of the spinal cord or the olfactory bulbs. Next, you have bipolar. So you have two distinct processes that definitely arise from the cyton, but the cyton will be at the middle, and you have one process from one end and the other process from the other end. And these are found in retina or olfactory epithelial layer, vestibules or cochlea. So basically ears, auditory visions and visual mission, auditory and visual information. Next, you have multipolar. So you have these different, these type of neurons. They have one axon here, but they have so many dendrons on the other side. So these are what you will find generally in the motor neurons, because they are longer. They have multiple avenues to receive information from, and that's what motor neurons are supposed to do. They're supposed to receive information as quickly as possible and react as quickly as possible. And then you have apolar neurons. These have no definite like cell process. So you don't have any particular cell process which is going on in a certain direction. They're almost spread everywhere. So that's why they're called apolar, and you will find them in hydra. So hydra, you remember, they have the most simplest nervous system, and you will also find them in retina, amacrine cells. Now, this was structural classification. You also have functional classification based on what kind of information they carry. So sensory, as we have already seen, they convert signals from the external environment. So they receive the cue, they respond to the cue, and then they give this information to the spinal cord and the brain. And that's why they are mostly pseudo-unipolar in structure. So if you see here, they have this information being received, and then they carry this information further. Motor neurons, they are multipolar. So if you see motor neurons here, they are this multipolar huge dendritic structure here. And again, motor neurons are the one that receive cue from the central nervous system and extend to the body parts. And then you have something called interneurons. These are also multipolar in structure, and they connect the sensory and the motor. Sensory receives, motor has to give it out. So sensory receives and send it to CNS. CNS is sending out information to the motor neurons, and they are both connected to each other by the interneurons. Okay, and lastly, we will talk about the nerve impulse. So how is this electrical activity actually taking forward in the nervous cells or nerve cell. So just to understand nerve impulse, the nerve tissue, the main property of nervous tissue is excitability and conductivity. So what does it mean? Excitability is the ability to enter into state of excitation which is in response to stimulus. Excitation here means, so in general condition, in resting condition, your any part of your body, so here for example in nervous tissue, they have a certain concentration of ions inside the environment, cell environment and external to the cell environment. And based on that, they have a certain potential. To get excited means to respond to anything by changing the concentration of your ions across this membrane. What happens? These ions are carrying charges. So you become either positively charged or negatively charged based on the stimuli. And that change that is happening because of the change in the concentration of your ions is called getting excited. So that's one of the features of nerve cells, which is excitability. And the next is to conduct this excited state or the potential across the information. So you have received the information, there is a local change in the potential, local excitation. This has to be propagated to its proper destination. So that is carried by the nervous tissue and which is called conduction. And this happens via the nerve impulses. So the nerve impulse generates and carries the information further. And so this is a huge network, right? So from one net dendron to the other dendron, or from one axon to the other, it is carried, like it is transferred at the synapses. We have read about synapses in the last session. So if you see here, you have the synapses, right? So you have one axon is carrying the information. This information comes at this end, picked up by the next dendron, and then this also carries this information further till it reaches the CNS. Now just imagine how fast it takes place. So you have, let's say you're walking somewhere and you happen to put your leg on a pin, which is very common example. Now you instantly remove your leg. But how did that activity actually take place? The moment you touched your leg touched the pin, the nerves, sensory nerves there gave the information to the CNS and the CNS responded via motor neuron saying that pick your leg up. So there were sensory neurons transmitting the information, motor neuron carrying the information. And we have seen how it happens in milliseconds. So all this long distance from your leg to your brain, let's say, how does it happen? It can't be one nerve which is carried so long, right? So all these happens via at these synapses when the information is carried by different nerves. So one nerve receives, reaches synapse, gives it to the next dendron, takes that dendron, take the information, give it to the axon, and that's how you propagate the information till it reaches the brain. So that's why it is so important feature for the nervous system. So as I mentioned, so there exists an action potential which is because of the movements of these ions, and these mainly are carried by the sodium and the potassium ions. And nerve conductors, they will have high membrane resistance and low axial resistance. It means that the conduction is quite high, not at the membrane, but so at the membrane they have high resistance. So the membrane will avoid the leaking of the ions, but axial means along the axon, it will have less resistance. The ions or the electrical impulse is not resistant, so it moves very swiftly through the axon, but not at the membranes. Then the electrical synapse has its, so just as well, just I mentioned that you have escape reflexes, you have the heart is going up, the heart rate is going up, your retina pupils are getting dilated. All these are very fast responses, and this happens because of such fast electrical impulses. So we'll read about the action potential now. So we'll go one by one. So we have a resting state, then we have excited state, and then we come back to normalcy. So we'll go very easily on this. So the first thing is when you are in a resting state or in a normal state, so the axon or the nerve fibers here, they are generally in the form of a cylinder. And here, this interior part is filled with exoplasm, and the exterior part here and the lining is called exolemma. So there are fluids both inside and outside of this membrane. Now, outside the axon, you have the negatively charged chloride ions, and the sodium ions, which are positively charged, they are neutralizing those chloride ions which are present here. Right, so you have lots of sodium ions on the outside membrane, while on the inside you have the potassium ions which are neutralizing these chloride ions. So chloride ions, so there is positive ions both outside and inside. Outside there are more of sodium ions, inside there is more of potassium ions. Now this membrane is, however, a little bit leaky. Leaky means, so this movement of sodium and potassium across the membrane is based on the permeability of the cell membrane, and it is relatively more permeable to potassium ion, because of which even at this stage some of the potassium keep going outside. Now because of here this at resting potential, you have a negative potential here inside. So that difference here is somewhere around 70 to 90 millivolts. So that is a resting potential electric volts, like 70 to 90 millivolts is what you will see in the membrane if it is at rest side, and it is still got polarized because there is certain negative charge to it, right? And you have these pumps, sodium potassium pump, as the name suggests that they will be involved in the movement of sodium and potassium across. They are what keeps this movement or this overall electrical charges at equilibrium. Now as I mentioned, so when you receive a Q, so the pump here which is placed on these axons, they actively pump the potassium ion and sodium ions. So what do they do? So when you have the so actively means against the concentration gradient. So these pumps, they push the potassium ion outside into the exoplasm, and sodium ions are pumped from the exoplasm into the exo. So sodium are pushed out and potassiums are taken in, right? That's what happening in the normal state. So, and this is what I mentioned a while back, that potassium ions, they have more permeability. So there is a loss of potassium ions, which is more than the gain of sodium ions. So sodium ions are not coming inside, but the potassium ions are, which should have been inside, they keep going out because of the leakiness. And that's why there is an overall net loss of potassium ions from the axon. And because there is loss of this positive charge, you have more at resting state, it is more of a negatively charged membrane. Now, so now let's say you receive a stimulus, certain kind of stimulus. Now at this juncture, the membrane will start getting depolarized. What will happen? The pump, which was actually functioning to put the sodium ion in and sodium ion outside and potassium ion inside, will now start functioning the other way. What will happen? Here the potassium ion will now rush outside the membrane, and the sodium ion will start taken inside. So let's just go back once again to understand the difference here. So in resting state, the pump was actively pumping potassium ion from outside. But now the potassium ion is being rushed outside. So the ion which was taken in is now being pushed outside because of the stimulus. So you have the potassium which is being pushed out, and sodium which was being pushed out is now being taken in. And this change in the concentration of the two ions leads to something called action potential, because there is a charge difference now, right? So these membranes are now called depolarized. They were resting, now they are depolarized. And this depolarization, now if you see here, because of this movement of sodium and potassium, it got depolarized. So we are in this stage now. Now this is localized depolarization. This has to move out. This has to move towards the other neurons so that they can relay the information. So the first thing, when this sodium is moving in, the negative resting potential is first cancelled out, because there was a certain level of deficiency in the initial part, right? It was negatively charged. So initial movement of sodium inside will first neutralize that charge, and once the equilibrium is reached, it is only after that that the membrane will become depolarized. And this happens at a difference of 30 millivolt. Means what do you mean? There is a certain level of sodium which will first enter, but this will not lead to depolarization, because this is negatively charged. The membrane was negatively charged. You are putting in sodium which are positively charged. So first it will neutralize these charges, make it without any charge. But then you have certain threshold which is crossed for the sodium ion. Now there are more of these sodium charges. Then the potential will differ, like change, and a difference of 30 millivolt is what is at the maximum concentration of sodium. So you have all the sodium inside now to have a potential of 30 millivolt. Now what will happen? So now the potassium pump will start to operate and now the action, so this will be then propagated. As and when this happens, the adjacent membrane opens up, start doing the same information, start doing the same activity. So this polarization changes, this pump gets activated, they start taking in. Now at the rear side, when this information is passed on to this side, these will start getting repolarized. Means the same step that was taken in the previous step will be reversed here. Here again now potassium will be pushed out and sodium will be taken in. But when you have reached the stage of repolarization, they start doing in the opposite end so that they bring that potential back to normalcy. So repolarization again will have the potassium ions will be rushed inside and sodium will be pushed outside, exactly what was done in the resting potential. And here then the pulse will stop. Impulse will now be not transmitted throughout the nerve fiber during the refractory period. So a nerve stops responding or stop propagating impulse when it is completely getting repolarized. So this is being transmitted. As and when it will reach the end, the other parts are getting repolarized. As in repolarization ends here, this will no longer transmit information because that's what exactly it was not doing. There was no depolarization to start the relay of this information. So this period when it is start gaining the reverse or gaining the original concentration of the sodium ion back is called the refractory period. So this is the normal neuron, right? This gets activated. This is where the initial repolarization takes place. This is propagated and carried forward. This is continuous conduction. But now, this is the other kind of neuron that we were talking about. What was this? There is this. This is called the myelinated sheath. So here you have these junctions, nodes of Ranvier. Now, these act as insulator. Means what? They do not, at these sites of the neurons, this kind of exchange of ions will not take place, right? So what will happen here instead is, because of this is called saltatory conduction, means there will be a jump across these nodes. So uh, here when the pulse is implicated, you can see that the movement will be sooner. So from here they will move here, then they will move here, then they will move here. So the information is conducted at a faster rate in a saltatory conduction than in a continuous conduction, and you need this in the brain to relay information faster. And that's why it is present in the brain and the spinal cord, so that information is conducted at a faster pace. And that's what happens. That's why you need the myelin sheath there. And then you have the synaptic transmission, which is at the end of these individual neurons, where it will pass on the information to the next neuron. So we have already seen part of this in the previous sessions, where we knew that the impulse comes here and then it releases acetylcholine and then it binds to the receptor or the next dendrite and then it passes the information. But what we'll see here is how the acetylcholine is released from the or in the neurons. So in this synapse here, a membrane of the telodendria. Telodendria means dendrons, which is at the end of the fiber. So telodendria. So you have a presynaptic membrane here, and then you have a postsynaptic membrane. So this is the presynaptic membrane, the cleft, and then the postsynaptic neuron. So when you have this action potential which has been transmitted across the fiber, when it reaches in this presynaptic membrane, here the presynaptic membrane becomes permeable for calcium. So that impulse that is relayed onto this part, it makes the membrane leaky or permeable to calcium ion. Now calcium ion, which was in the outside region, it enters this membrane, and here when it comes inside, the vesicles which have the acetylcholine molecules, they are burst open. When these are burst open, you have the you have these acetylcholine vesicles going and binding to the edges and then bursting open. So the acetylcholine are then released into the cleft. These acetylcholine, if they are, let's say, that is signal for a next muscle cells or any other cell which is have a receptor, they can bind to the receptors there and then affect the information. Or if it is a neuron, it will bind to the receptor and then generate the impulse for that further information on the relay. So that's how you see how the information was carried from the nerves to the next nerve via synapses, and these nerve fibers and nerve cells and glial cells making up the nervous tissue making up the nervous system, which consists of the brain and the spinal cord, and at last the nerves which actually are responsible for carrying out the information. So now we'll go over some questions from the previous year's assignments and try to solve them. This should have all the information that we have just discussed, but even if you have some difficulty, let me know. So the first question is the germ layer which gives rise to central nervous system. So we have discussed this, which layer gives rise to the different organs of central nervous system. Next is the triple membrane of connective tissue that protects the brain from external pressure. So what are the layers or the outer coverings of the brain called? Next is the two hemisphere of cerebrum are connected with nerve fibers called. So what is the nerve fiber? What is the connection between the two hemisphere of cerebrum called? And the next is the involuntary actions are controlled by which of these which of these parts. So just try to guess, like remember maybe what we had discussed. Yeah, so this is the answer. The first is that the layer which gives rise to central nervous system is the ectoderm. The protective layer of the brain is called the meninges. It was carpus callosum, which was the connection between the two hemispheres, and the involuntary muscles is being controlled by medulla oblongata. This would have been easy if you just could connect with the last session where we had discussed in detail how heart skeletal muscles are controlled. Okay, the next questions are the area deep in the brain where pineal gland is found. So this was either thalamus, epithalamus, metathalamus, or diencephalon. Next, the primary control center for reflex action again is present in which of these regions? Next, the nervous system that maintains homeostasis of internal organs and initiates stress response is which region or which system? And the next is the facial nerve originate at which of these sites? So this also we have discussed. Which is which of these part is responsible for facial expression? Okay. So the answer to the first question is epithalamus. We had discussed this where we find the pineal gland. The second is spinal cord, which is responsible for the reflex actions. Then we have the autonomous nervous system, which is responsible for maintaining the homeostasis because this is responsible for all the internal organs and the glands. And then the facial nerve originate from pons varolii. We had seen that it is responsible for the regulation of facial expressions. Next, the electrical activity of the brain is this is easy. This is known as what or can be known from which of these activities? The number of thoracic spinal nerves in the humans are what pairs? We partly discussed this. Next is the membrane that binds to neurotransmitters and responds via depolarization or hyperpolarization is which one of these? And last is the neurocytes without dendrites and axons are called what? Are found where actually, sorry. Okay. So the first part is EEG, electroencephalogram. This should have been easy. The next is the number of thoracic nerves was 12 pairs. Um, the response to the membrane that responds to neurotransmitter but not to if it would have been an electrical impulse which is responding, the membrane which is responding to an impulse, this would have been presynaptic, but because we are talking about neurotransmitters here, this would be postsynaptic. And the next is the neurocytes without dendrites and axons are found in the amacrine cells of the retina, where we had discussed at the end of the apolar neurons. Next, the structures in neurons, which is formed by coiling of endoplasmic reticulum around ribosome, is which of these? And we have discussed this. Next, the leakage of ions in neurons while traveling down the axons is prevented. This I have discussed like multiple times now. In which of these layers? And next, the type of nerve conduction from brain to muscle is carried by which of these neurons? And the principal conductance which is not responsible for maintaining the resting membrane potential is which of these ions? So basically it means that which of these ions are not responsible for maintenance of resting membrane potential. Okay. The first one we had discussed, it was Nissl bodies. Second was new dilemma, the layer with this quant cell, myelin sheath. Third, the brain, from the brain to the muscles. So it is the effect, so efferent neurons. And the last was calcium, because we had discussed about sodium, potassium, and chloride. Chloride was being neutralized by the other two ions, sodium being mostly in the outside region and potassium mostly in the inside region. Calcium we had discussed when we were talking about transmission at the synapses. Next is the resting membrane potential of neurons is at which range? The ion responsible for the increase in positive ions and dropping the negative potential inside the axons, which leads to the generation of electrical action potential. Let's just take a little time, read the question, and try to understand it. Next is the period during which membrane cannot receive any other impulse. This is the last step or last stage. So, what is that called? And the next is the small gap at the end of neuron, which allows the signal to transfer from one neuron to other, which is also should be easy. Yeah. The first one is minus 50 to minus 75. We had read about 90, but it's a range, so we generally consider it from minus 50 to minus 75. Next is the ions which move inside and leads to the action potential. So that is sodium. Sodium is moving inside. Next is refractive period. It is a period during which there is no impulse which is being received by the membrane. And the last is synapse. So that's it. That ends the questions that we I could compile from the last assignments. But if you have any doubts regarding this topic or anything related to today's topic, you can post it in the group in the group in my video. If you have any doubt, let me know. Thank you.